course, this is incredible selection for the pathogen to evolve away from recognition. And so the new strains that have emerged are called race 1 strains, and these have loss of function of these two effectors. And so they're not recognized by the resistant genes that are present in cultivars. And these are now prevalent worldwide. So there's a real issue in terms of trying to identify resistance to these other strains. And so Jana was interested in trying to develop a seedling-based assay for resistance. So typically these assays are done in adult plants, and tomato is much larger than Arabidopsis, and so we were trying to see if we could make this a little bit more Arabidopsis-like. And so if we did it on seedlings, we could make this much higher throughput, have really large sample sizes. The growth time would obviously be much reduced, so we could require less space for them as well. And we were hoping that we would have more discrete phenotypes, so we'd remove some of the variability that's seen in adult plants. But obviously, we would have to still be able to show typical resistance and disease that would be seen in an adult plant. And so we decided to test and validate the system with cultivars that had or lacked the PTO-PRF gene cluster, and use DC3000 as the strain that would either be recognized or cause disease in those backgrounds. So the basic setup of the assay is that we would flood inoculate tomato seedlings and incubate them under high humidity. And we were hoping we would see really discrete phenotypes. So we would have resistance, which would be seen by healthy plants, and disease would be death of the seedlings. And then once that was established, we could then use the natural diversity in tomato to try to identify resistance to these strains. Okay, so we first had to establish a system that could work. So here we have the susceptible cultivar lacking that cluster versus the resistant cultivar. And we had to optimize for the optical density of the bacteria that would cause a difference in phenotype across the two genetic backgrounds. And so here you can see in the susceptible background, the leaves are yellow and there's some speck, which hopefully you can see at the back too. In the resistant plants, the plants are really green and healthy even though they've been infected. And we were able to quantify the level of bacterial growth in these plants. So here we have log growth on the y-axis, across optical density on the x-axis. And in gray are the susceptible plants, and in black are the resistant plants. And we got about a 2-log reduction in bacterial growth for all of these different optical densities. So this confirms that the resistant plants are supporting some level of bacterial growth, but it's not sufficient to actually result in disease. So we wanted to also see other common measures, whether we could see those in seedlings. And so here again we have susceptible or resistant plants, vacuum infiltrated with magnesium chloride as a control versus our infectious strain DC3000. And so here in the susceptible background, at this early time point, we don't see disease. So this is pretty typical of these assays. If you take a very early time point, you won't see disease. You have to wait a few more days to see that. However, in the resistant background, you can see that the leaves are starting to shrivel and crinkle, which is, again, characteristic of the hypersensitive response, that macroscopic phenotype. And we could, again, quantify this using ion leakage. So here we have ion leakage on the y-axis and time on the x-axis. And in the resistant plants inoculated with the pathogen, we get much higher, more rapid increases in ion leakage compared to the susceptible plants. So this gave us a lot of confidence that the system was actually working, that we could distinguish resistance and susceptibility, we could quantify that, and we could actually visualize the hypersensitive response. So we then went on to the T1 strain, which is again one of these emerging strains. This was a kind gift from Vita Coker's lab at UC Davis. And so we tested again different bacterial optical densities, and you can see we needed a little bit more bacteria here to be able to get complete death of the seedlings. And we didn't have a resistant control here because obviously this is what we're looking for. So, and then we then screened sections of wild tomatoes from South America. And so Charlie Rick, who at the UC Davis has a huge collection at the Tomato Genetics Resource Center of accessions that you can order. And then these are naturally diverse, so they're present in different geographical environments. They've been exposed to different pathogens, and we use these again as a resource of genetic diversity to look for resistance. And so Jana carried out a screen of 96 wild accessions and identified 85 that had complete death of the seedlings and 11 that had at least one resistant seedling. And then she tested the progeny of these resistant seedlings for resistance to make sure that the resistance was heritable. And for 10 out of the 11 accessions, we saw resistance, but you can see that these are genetically complex. So some of them had very low levels of resistance up to almost 70%. So these are two of the strongest lines. So we have a Solanum neoricki and a Solanum habricades. Again, on the left side is the magnesium chloride inoculated control, so the plants are green and healthy. On the right side is the infectious strain. So on the cultivar, all the seedlings die, but with the infectious strain on these two wild accessions, you see there are individual seedlings that are surviving. And to make sure that these were really surviving, we transplanted them off of the plates, grew them up to be adults, and they did not show any sign of disease. And you can see across the population, we had 63 to 68% resistance for both of these wild accessions. So we then wanted to quantify the level of bacterial growth that would be supported in these backgrounds. So here we bin the seedlings by phenotype and then calculated how many bacteria were able to grow. So again, here we have bacterial growth assays with log growth on the y-axis. And in the susceptible wild accession or the susceptible control, we saw very high levels of bacterial growth and about a log reduction in bacterial growth in the resistant wild accession. So far, all of this has been done in seedlings. We then, of course, wanted to make sure that we'd still see resistance in the adult plants. So here we would dip inoculate the plants and incubate them under high humidity. And again, either look for resistance, so leaves that looked basically untouched versus leaves that had bacterial speck. And so here on the top, you can see the cultivar, which is heavily diseased, lots of bacterial speck, versus the wild accession 1329, which was asymptomatic or had very mild symptoms. And again, we could quantify this difference. So in the susceptible background, we have about log 6 growth, and then about a log reduction of bacterial growth in the resistant accession. So Jana's continued this screen and has identified multiple resistant lines in addition to these two that I've already mentioned to you. And I'll just take a few minutes to tell you about one of these lines, which we call SPA for line A of Solanum pimenillifolium. Again, this one has individual seedlings that were able to survive resistance, but across the population it has almost 90% resistance. So we thought that this one might be a little bit easier to work with. And interestingly, this line also exhibits that macroscopic immune response to the infectious strain that I showed you before with the isogenic lines. And so we were really excited about this because this is a really nice scoreable phenotype that can be used to actually look at effector recognition. And again, we can quantify this. So Ilea did tremendous work to try to get the quantification to work here. And so we have this really nice curve of the wild accession with the infectious strain in comparison to the magnesium chloride control. And you can see that we have really elevated levels of ion leakage in resistant plants. And again, this is associated with a reduction of bacterial growth. So we have about 2 log less bacterial growth in the resistant background. And so Jana and Ilea are now really focusing on this line to try to identify which genes are involved in recognizing these specific effectors, but also what else in the strain is triggering resistance. And so Ilea, for her dissertation work, is trying to really identify which are the bacterial proteins that are important in triggering resistance. And so she's creating knockouts in the strain to look at whether there are conserved bacterial proteins that are important or effector proteins. And she's also looked at the present in T1, which is this emerging strain. And we looked in particular at ADR RPT2, which Brian's lab has done a tremendous amount of work on, and this is not recognized in tomato. We also have looked at HOPAS1, which is also known to confer resistance in other systems. And we have weak resistance in tomato, but we don't think that this is the cause of the overall resistance that we're seeing. And so ILEA is continuing to test other effectors here to see if we can determine which effector is actually responsible for the recognition. And then she's also taking unbiased approaches to try to figure out what else could be recognized if it's not an effector. And so in collaboration with Joe Bondi-Dinami, we're doing CRISPR on effector clusters. And then in collaboration with Adam Deutschbauer, we're also doing TNC to try to identify those other molecules. Okay, so in this part of the talk, I've told you about a seedling flood assay that we developed that can recapitulate measures of both susceptibility and resistance. We've identified multiple wild accessions that have resistance to this race 1 strain of Pseudomonas, and we're in the midst of characterizing the resistance and then also identifying what molecules in the bacteria are responsible for that recognition. And so these will be important tools for plant breeders for resistance. And our interests in particular are trying to identify the genes that confer resistance and then trying to understand, again, the molecular mechanisms of resistance in those systems. Okay, so today I told you about 2 stories on the immunity side of the lab. And so the first one focused on molecular mechanisms of immune activation and trying to understand what is important for resistance in that context so that we could potentially engineer resistance. We've developed new tools and approaches in both, and we're trying to identify what in the bacteria is important for recognition. And then we're also really interested to identify sources of genetic resistance in tomato that could be used in applied contexts and also try to get to the molecular mechanisms of recognition. And so altogether, we hope that this work will allow us to really be able to more systematically engineer immune receptors so that we could have resistance against multiple effectors or multiple pathogens, or potentially also identify new strategies and targets for resistance. And this is a really important problem because crop losses from pathogens are estimated to be $220 billion. And with the projected increase in the world population, we need all the food that we can to be able to feed everyone. So I want to thank the people who did the work. So Maëlle is the one who established the transient expression assay and has done all the molecular work trying to understand the SARS-Z interactions. Jana established the screens in tomato and identified those candidate lines. ILEA is working on the bacterial side of those tomato infecting strains and identifying the molecules that are important. And Carl and UN, I couldn't really talk about their work, but they also have really interesting projects. We've had the fortunate help of a lot of great undergrads in the lab. And Andrei and Eliza in Romania helped us with structural modeling of ZAR1. Chelsea and Shodan help us with the ecotype screen. Joe and Valent are helping with the CRISPR approaches, and Adam and Morgan with the TNC. And then our funding is from the USDA and NSF. So with that, I thank you for your attention. I'd be happy to take any questions.
Could you talk a little bit more about how you would engineer broad spectrum resistance? Is it by expressing the various Z1 forms that are for engineering, new versions of ZAR1 to interact differently with these, or what's
so for the examples I showed you, it's the same ZAR1 protein in Arabidopsis that can interact with multiple different receptor-like kinases. That are all genomically clustered. And so they're actually, they're relatively similar to each other, but they're not identical. And so part of what we're hoping to do with the diversity screens and the error-prone screens is figure out what those determinants of recognition are. So right now we don't know, for instance, whether there are multiple changes that could occur that would all trigger resistance, and if we could make a pocket of the kinase that would be particularly sensitive to a pathogen, then that could be like a sort of mimic of a decoy, so that it would, we could use that as a way to detect that an effector is present. So in our case, we have acetylation of the kinase. The other group has uridylation of the kinase. The third group, they haven't shown it directly, but for a homologue of that effector, it's an ADP ribosyl transferase. So there's lots of different things that could happen to that kinase that would presumably change the conformation of it, and that would trigger resistance. So at this point, we're still kind of in the discovery phase of figuring out, what are those critical residues that might distinguish between recognition or no recognition.
So I have a question about the role of acetylation, because in a couple of your assays, right, you didn't require acetylation. The acetylation deficient, right, Z1, and then also, anyway, there are two different assays that didn't require it.
Yeah.
So I was wondering if it was maybe less about acetylation per se and more about stability, like actually stabilizing the protein.
Yeah, so I guess that's possible. So one reason that we think it's acetylation is that if we have the catalytically inactive effector, we don't get recognition. So that would suggest that it's more about whatever the effector does to the kinase that's important. Our interpretation of the acetylation data is that there are additional acetylated sites in Z1, and we have been interested in finding those, but we have not been able to get complete coverage of the protein. So when we identified those two acetylated sites, we have about 60% coverage of Z1, and trying to get the right digest to cover the rest of it has been very problematic. So we think that there are additional sites that could be acetylated, but there are so many serines and threonines that we have not identified those yet.
So, sorry, so when you, I mean, that totally makes sense. I couldn't, you had a lot of data in there. So when you when you inducibly express the Z1 and the ZAR1 without the effector, I thought you still saw the interaction and the response and the HR, but did you just see the interaction but not the HR?
We see the HR when we co-express HOPC1A and Z1. We don't have to have ZAR1 to get that recognition in a non-silence plant. And the acetylated or the acetylated mutant can still interact with ZAR1. Yeah.
I just have a quick question about the tomato part. Are the wild isolates, can they be interbred with the modern cultivars?
They can, yeah. And so not all of them.
And
but
the one you care about most that has the most
yes,
reason?
yeah. So yeah, it's a Solanopyranilfolium, and that was historically used to cross into cultivars, so that one is not a problem. There are some tomato that are not able to interbreed with themselves, and so they're very genetically complex, so we've intentionally avoided all of those, and we've stuck with the ones that are able to self-fertilize so that they'll be a little bit less genetically complex. But yes, they can definitely do that. Yeah, cool. Yeah.
I also have a question about the tomato, which I find super interesting. What is the susceptibility or resistance of this tomato to other tomato pathogens? So like Fusarium, for
Yeah.
example.
We have not tested that. I mean,
And also to other bacterial treated in tomato, because if you are suspecting a pump, there might be something common maybe to other.
sure, and that's possible. We have really not gone down that road. We've kind of focused pretty exclusively on this strain. I think it would be interesting to test that. We would have to, and we think it has the possibility of being extended to other bacterial pathogens. They would just have to be optimized. We'd have to, again, reset up all the assays for every different pathogen to make sure that it could behave, the way we expect it to. But I think it's a possibility. Right now, I think we have our hands a little bit full trying to map the resistance that we have, but down the road, that could be something we explore as well. Yeah.
So the pseudokinase is acting as, you're saying, as a decoy. Presumably there's target?
Yeah, so the effector is the acetyltransferase, and the pseudokinase is the decoy, we think.
Oh, so I was just wondering, is there any, like, can you do sequence comparison? Can you use the pseudokinase to predict targets?
Yeah, so we have definitely been interested in doing that. So back to the first part of your question. So we do think it's a decoy for the real virulence target, which we would assume is a kinase. So we've been very interested in trying to figure out what that target is. It has been very challenging. So this kinase only has a kinase domain, and we've had a lot of trouble finding something that's similar enough, but is actually the target. And all the kinases that we've tested so far are not targets. HOPC1A is also known to have a lot of different virulence targets. So there have been multiple groups that have shown like tubulin is a target, and enzymes involved in JA biosynthesis are targets. So it probably has multiple modes of action, and the kinase target is one of those potential ones. Yeah.
Is the nucleotide binding part actually binding nucleotides, and if so, what time and where?
Yeah, so this has been a big question in the field for a long time. So there's one group that was able to show that they were able to bind ATP or ADP. Doing those assays has been very challenging. Most of these proteins are not soluble, and so trying to purify them and do those tests has been very difficult. So we assume that it's going to exchange ADP and ATP, but we haven't been able to show that directly. And as I said, many other groups have also tried that and not been successful. So it's still an open question.
Yes. As someone outside the field, I'm very impressed with the multiple capabilities that plants have to defend themselves against a myriad of pathogens. In your opinion, if we look at plants in general and just take bacteria, what would be the major mechanism that plants in general have for defense against these organisms?
Well, so as of right now, I've been mostly focusing on inducible immunity, but of course structural defenses are a big part of that too. So if plants can prevent pathogens from even getting into the plant, then you don't see disease. And so those would be things like the cell wall, the cuticle, other things like that. So that level of immunity can work against a lot of different pathogens. The inducible immunity is important kind of for when the pathogens are already getting access to the plant. And then it's a matter of, there's lots of different pathways that can be targeted. I mean, they have basically evolved every possible strategy to overwhelm resistance. If it's, like these stealth pathogens where they kind of fine-tune, make little changes, or other pathogens that just kill the host and eat it. So I wouldn't say there's exactly a common theme for those.
If we look at the cell wall type,
Yeah.
can plant breeders improve on that?
Well, perhaps.
Has anybody done it yet?
Well, I mean, I think people have tried, to look at mutants in cell wall biosynthesis and see either would they be affected in resistance. They're usually focused on like a particular plant pathogen interaction and whether it has an effect there. I'm not sure that anybody's tried to engineer cell walls in general.
That would seem to be the most promising, if the investigator lives long enough.
Yes. that could be a
Actually,
challenge.
the comments along that way, I mean, some cell wall biosynthesis enzymes are actually induced by pathogens. In a sense, it's also part of the inducible immunity.
Yeah. But as with most cell wall things, it's complicated.
Any other questions? If not, let's thank Jane.
I just get my